All right, well, thanks very much. So what I do, I'm a professor of biomedical engineering at Boston University. My background has been in applied mathematics, fluid dynamics, applied mechanics for many years at the University of Illinois, which is where my son David, who you see up behind me, was diagnosed by my wife, a pediatrician, as an infant with type 1 diabetes. That was about 16 years ago, so he's a rising senior in high school now. And as professional academics and a physician, we could take good care of David, but the realization that one day he would go off to college and have to manage his own diabetes was really quite daunting to me. We checked his blood sugar 15 times a day when he was an infant. We got him on continuous glucose monitoring about 7 or 8 years after that. He wears an insulin pump and has since he's been 13 months old. And I call him the boy who lived, as I do every person who's been diagnosed with type 1 diabetes in the last century since the discovery and purification of insulin. So it's a life-saving medication, but ultimately it's an extremely difficult drug to dose. And it's extremely dangerous. It has a very high toxicity index, a very narrow therapeutic range, which essentially means, obviously, if you overdose a little bit, you can kill somebody. And nevertheless, this is a drug that everyone has to dispense who has type 1. There's just no getting out of it. You literally cannot survive without this drug. And we've done a great job taking care of his blood sugar, but it's a totally different challenge when your son has to go off to college and manage it on his own. Can't do that overnight. Your blood sugars are typically lousy overnight because you're afraid of low blood sugars. So it was really soon after he was diagnosed that I thought, as an engineer, we could solve this problem, at least for type 1 diabetes, of managing glycemia. And it really is, type 1 diabetes is an engineer's dream. This isn't so much a drug discovery problem as it is an engineering solution, because what we've learned from this very large-scale multi-center study in the 1980s called the Diabetes Control and Complications Trial is that if you can maintain blood sugar levels very near the normal range indefinitely, in perpetuity, you can stave off all long-term and acute complications of type 1 diabetes. And that really is an engineer's dream. It's a dynamical control process that you're trying to master. The tools were not quite on the table in 2000 when I started working on that problem, but I had a PhD student helping me work on the mathematical algorithms, which ultimately we took to pig studies when we moved to Boston University, and collaborated then with folks at the Massachusetts General Hospital to begin clinical trials about 8 years ago. First in the inpatient setting at the Massachusetts General Hospital Clinical Research Center for about 4.5 years, and then ultimately to outpatient studies running on an islet. On an iPhone system, we built this, cobbled together an iPhone with insulin pumps and continuous glucose monitoring, and we could achieve glycemic control never before achieved, both in adults and children, and specifically adolescents and pre-adolescents in real-world outpatient home use settings, summer camp settings where there was no restriction placed on diet or exercise, with nothing more input to the system other than the subject's body weight, and then it would automatically adapt and adjust insulin dosing to the subject's ever-changing insulin needs. It's the epitome of a personalized medicine right there in your pocket. Could we just advance to the second slide? And that's all I really wanted to show. So what we have done over the past couple of years, and I think this is really apropos this session, is to use the resources that Boston University has for all kinds of things, nothing to do with translational technology, as well as things that do, to our best advantage. We raised $2.5 million in private giving from mainly parents of kids with type 1 diabetes like ourselves to build this technology. So we've now fully integrated what runs on an iPhone and what runs on with these little pumps and continuous glucose monitors into the standalone device. We call it the islet in homage to the islets of Langerhans. It's about the size of the old iPhone. Remember these things from 2007? About the same size, maybe about 5 millimeters thicker. But this is literally, carrying your glucose metabolism in your pocket. This whole system has 2 pumps, one to deliver insulin to lower blood sugar and glucagon to raise it, integrated continuous glucose sensing, and of course all the mathematical algorithms we've developed and been testing in almost a decade's worth of clinical trials are embedded in firmware on this device with a very simple user interface of our own design. We built this as academics using contract research organizations, and so what I'd like to talk about when we get to the Q&A is not so much what the impediments that academia may have put up in front of us, or even the funding and so forth, but rather the opportunities that come from a major research institution, the way you can use that institution in novel ways to produce something like this that allowed us ultimately to spin out a small startup company, which we call Beta Bionics, that will ultimately commercialize this technology.
That's a great pun for the name of your device. So, Alison.
Just trying to.
Yeah.
There.
Yeah, I think we're good. Go for it.
Okay, so continuing on the theme of personalized medicine or precision medicine is not just genomics, but includes a lot of other areas of engineering. I want to talk for a little bit about congenital heart disease and cardiovascular surgery. So many of you may not know that congenital heart disease affects about 1 in 100 babies. It's actually far more prevalent than childhood cancer, although that tends to be the thing we hear more about. It's the leading cause of infant mortality in the US. But it's a challenging area to advance in terms of treatments because while it does affect 1 in 100 patients, there are over 40 different types of congenital heart defects, and you can have any number of combinations of those, and so every patient is unique. And surgeons have to often make sort of on-the-fly decisions about the best surgical design or approach to use in the OR for that particular patient. So the conditions are rare and varied. There tend to be very small patient cohorts. And so again, echoing some of the things we heard earlier, the design of clinical trials can be challenging. And also following some of the other sort of rogue diseases, the market sizes are very small. You see a lot of off-label use of devices, for example, ventricular assist devices or stents, where people are having to make the assumption that a child is a small adult, which is not actually the case. And then, going back to how did we start treating these patients surgically? So actually the very first heart surgeries were performed on children with congenital heart defects back in the 40s and 50s. So the pictures that you're seeing on the slide are some of the first teams that performed these surgeries. This really required efforts on the part of sort of maverick surgeons who were willing to try very out-of-the-box ideas on patients who had conditions that were uniformly fatal if they didn't do anything. And a lot of this, continuing to the present day, now we've sort of arrived at methods for treating some of these patients. For example, patients with single ventricle heart defects who were born essentially missing half of their heart. We have treatments for these patients, surgical methods. They're still imperfect, and now we've hit a point where the advancement in surgical methods has slowed. Really, the surgical methods we're using to treat those patients, at least in the first stage, has not really changed since the picture that I'm showing here. So what we're trying to do is bring a more predictive or quantitative approach from engineering using computational fluid dynamics, much as we would do in the aerospace industry, to accelerate, say, the design of a new aircraft using simulation methods and bring that to bear on cardiovascular surgery. So what we do is called patient-specific modeling, and we typically take image data like MRI or CT data from a patient. We use that together with computational fluid dynamics methods where we know how to simulate blood flow. And we can use that to perform virtual surgery. And so, for example, we can use simulations as a test bed for novel surgical approaches. What you're seeing here is a comparison of a patient-specific model preoperatively and then postoperatively with two different alternate designs, one of which is what they do now, and then the one on the bottom is something that we are proposing as an alternative method, and we can then assess these using simulations at no risk to the patient, at least at this stage. So again, the goal is to enable this kind of quantitative decision making. This, of course, requires a team approach. So we are very lucky at Stanford to be co-located on the same campus with a leading engineering and medical school. But this requires really a team of engineers, surgeons, cardiologists, biologists, radiologists, all working together. So some of the things that I think have led to aid in our success at this is, being co-located on a single campus, having the presence of leaders in multiple fields, but also having leaders who are buying into this kind of new, more quantitative approach, and then having interdisciplinary, not only teams, but hiring. So I'm actually a mechanical engineer who's appointed in the Department of Pediatrics. So that requires some institutional buy-in. And the other thing that has helped our success in this sort of rare disease area is using open source software, where we work with other teams of researchers on common software tools, and some of those are listed here. These are just some examples that I'm showing. This is a novel surgical method that we developed for the third stage of single ventricle heart repair called the Fontan. And this was something that we were able to design and optimize in simulation and then take into a pilot clinical study. It's not always that simple. Another case on the bottom here is one that we're proposing in simulations. We've done some in vitro testing. We've done some animal testing. The animal models are very imperfect. And we're now at sort of the stage where somebody's going to have to take a leap of faith and actually try it clinically in order for us to advance. And in that, we're sort of taking an in silico, in vitro, in vivo approach to engineering. Some of the challenges that I see are in sort of validation. How do we collect the clinical follow-up data that we need? Ethical concerns, of course, in dealing with children and data collection and follow-up. In the area of single ventricle patients, there really is no animal model for this. And so you're forced to, in a way, the simulations are the only thing we have currently, together with the experiments, to test these methods. But then how do we make that rather large leap into the clinic? I mentioned the institutional buy-in, and then the sort of leap of faith that I think is still required on the part of a sort of a maverick surgeon to take that risk. So I'll pass it along.
Thank you.
Incidentally, that would have been amazing through my training, educational tool. We learned about all these procedures, and I never could understand them because I guess I don't have a very good three-dimensional visualization, but there was no way that we could actually understand the way these procedures worked without that. So that would have been a great application. So onwards to our next presentation. Professor Anderson.
Thank you. Great. Thank you again for the chance to talk. And so, as I considered the name of the session, I thought it would first sort of be helpful to give you my perspective on where therapeutics were not so long ago. And this is one classic example in my mind. It's the small molecule pill. Many of us take a couple of this particular pill, aspirin, every night. And certainly to me, this really defined therapeutics for decades. But if you look at, just for example, last year, one of the single most valuable drugs in the world is now a protein, an antibody. In fact, Humira sold $14 billion last year. And in fact, the biotherapeutics has completely changed medicine already. And I think it's not hard to see that along with these changes come complexity. These aren't synthesized, these are grown. And they're incredibly complicated three-dimensional structures that are therapies. And so, already engineering is having a tremendous effect on therapeutic discovery, but to me, it's really just the beginning. I think as I look at least at the therapies that I'm most excited about, these things start to look simple. And I wanted to give just a couple examples of the kinds of therapeutics that I think we might see over the next decade and perhaps even sooner. So one of the areas I'm particularly excited about are nanoparticles. These are biological nanoparticles. These are virus particles. And so historically, you're looking at these as something that's going to make you sick, right? But today, there are patients who've had dramatic cures as a result of using re-engineered nanoparticles, whether to address immune system disorders. There's exciting data with genetic disorders of the liver, really incredible results with the eye, an amazing range of diseases that are now going to be treated not with single proteins, which are complicated, but groups of proteins and other molecules. In fact, there are synthetic nanoparticles now that are showing utility in humans. There are lipid nanoparticles that have been developed to deliver siRNA to patients and have already been shown in humans to specifically turn off genes for an extended period of time in the liver. And, you know, I think, again, this is just the beginning. I and others feel that it may be possible to use nanoparticles or combinations of nanoparticles to change your DNA in therapeutic ways while you're still using it. And to just sort of put a visual to this slide, perhaps we could build nanoparticles that you could inject into the blood. Say you have a disease of the liver, and that's why I've drawn this liver green. Perhaps you can build nanoparticles that can specifically travel through the bloodstream and reach those disease cells, and then actually go into the cells and go in and actually change the DNA. I'm most excited about the CRISPR-Cas9 system, but there are other nucleases that are already being tested in the clinic. And then, you know, by direct modification, actually make enough repair events to permanently fix your DNA. And, you know, this was one of the things that actually made me come to MIT was this exciting potential. It took a long time for scientists around the world, including George here and others, to figure this out. But I think it's now really within our reach, and we've already started to see exciting results in animals that we think are going to translate. But, you know, I think this stuff is complicated, right? Imagine making specific nanoparticles that are designed to change your DNA, and the engineering needed to actually design that and make sure you can reproducibly make these, you can test these, they're going to be safe, they're not going to cause changes to the wrong piece of your DNA. I mean, there's some real complexity here that I think is going to require clever engineering. And again, in my mind, I think this is exciting and complicated, but it gets more complicated. So, you know, I think there's growing excitement around the idea that living cells can be therapies. I think as I look from the Cancer Center at MIT, some of the most exciting therapies are actually re-engineered T cells. The idea that you can take T cells, change them, and engineer them to specifically attack a patient's tumors and then put them back in as really smart missiles. And there's been some incredible results in various types of cancer. We haven't cured all of cancer, but we've seen some results that for the first time are really giving us a lot of hope that these sort of approaches could really change the face of cancer therapy. There are colleagues of mine like Doug Melton who have, through a heroic effort and combination of engineering and discovery, figured out how to make stem cells into islets. And perhaps, you know, he may be able to make enough islets to treat all of the diabetics around us. And again, this is complicated. It takes engineering. It takes engineering approaches to really make reproducible, pharmaceutically suitable batches of material that you can use to treat people. And again, this is complicated, but it also isn't done. It gets even more complicated. How do you take an injectable therapy or a pill for a macroscopic injury? I mean, maybe in the future we'll figure out how to re-engineer some salamander genes to help us just regrow limbs. But in the meantime, I think people like Jay Vikanti and others are really making progress doing tissue engineering. So combining engineering approaches with materials, with living cells, and actually coming up with ways to make macroscopic tissues that can be used in people. So this is an ear, as shown on a rabbit. But in fact, there are some three-dimensional tissues that are making money, not a whole lot, things like tissue-engineered skin. But I do feel, you know, there's real potential for three-dimensional macroscopic engineered tissues to be used to treat a range of diseases, and not just these superficial cosmetic ones, but in fact, things like perhaps we can build completely new organs to treat different disorders. So I wanted to end with just a couple comments that are specific to what I'm most interested in. One area that's really changed as a result of engineering has been the development of materials that are smart. And so historically, when scientists and doctors develop materials for medicine, they often use off-the-shelf materials. And here are a few examples. For example, the artificial heart. The doctors knew they needed something that was flexible, and what's flexible? A lady's girdle, and so the first artificial heart was made of the same material as a lady's girdle, polyether urethane. And you know, this trend really drove important advances in medicine, but it also became clear that these off-the-shelf materials were not perfect. And in fact, polyether urethane is nice and flexible, but biologically it can cause blood clots, and those clots can go to the brain. And so a real revolution in this field has been the engineering of materials themselves to make medical devices, whether they're nanoparticles or macroscopic devices. Briefly, one material that many of you, I'm certain, have had personal experience with are degradable sutures. These are smart sutures that when they get put in your body, they slowly dissolve over time. Here's a more recent example of biodegradable sutures. These are shape memory sutures where when you shift the temperature from body temperature to room temperature, room to body temperature, they take on a new conformation. And so you have one suture that takes on the conformation of a screw when it's shifted into body temperature, and you have another suture that's actually tying itself into a knot as a result of just a temperature change. And so I think what we're going to see ultimately is really the combination of engineering advances across many disciplines, whether it's engineered materials, engineered cells, the combination of the both. It's going to get complicated, and I think that's going to require a real investment in engineering and medicine. And we just had a couple questions we were given at the end to address and think about, and in the interest of time, I'll probably go through these quickly. You know, the first point is that we need to do this. It's getting complicated, but we need to figure it out. And you know, multidisciplinary training and research is essential, but there are still some questions around how we're going to fund it and how we're going to train people. An example is, so I got my PhD in genetics, but I'm a professor of chemical engineering. And there aren't very many places where a department of chemical engineering would be comfortable with a genetics degree. There are some that are and some that aren't. And so I think there's still a continuing debate around how much multidisciplinary training is right and how much really focused training is right. And that's something we need to figure out. And I think with that, I'll just end there and pass the slides along. Thank you.
All right, George.
Okay. I really like this topic of will engineering disrupt therapeutics? And I'll change it a little bit to how will engineering disrupt therapeutics? There's no doubt that it will, and I like and focus on the word disrupt. Daniel has just given us kind of an overview of the sort of things that his lab and my lab and many labs work on that could be part of that, but how can we really imagine it? And so I think back to when I was learning computers. I would build a 3-bit computer, which seems like a very limited one, and then slowly worked our ways up. Now we're working with terabytes and zettabytes. But what happened is you start building, there's a revolution in miniaturization and in new ways of encoding information, such that you now have, when I think of a small molecule like aspirin, I think of that as having a small number of bits. It has a small possibility of programming it. When you go to proteins, you have now larger number. When you go to viruses, even more, into cells, even more, into organs, potentially many, many gigabytes of information to handle. But the revolution that we've seen in this, in the computational and electrical engineering field has had to do with radical reduction in cost. The reason, you know, when we think about what's been disrupted, Uber has disrupted taxis, the newspaper industry has been disrupted, industry after industry is disrupted by essentially close to zero costs. You don't buy maps anymore. You don't. You know, GPS is assumed to be free, and so how do we bring this in alignment with therapeutics, where we've seen a steadily increasing cost of therapeutics over the years? And in particular, the one that I'm supposed to be representing, gene therapy, has the one approved gene therapy after decades of work and now 2,200 gene therapies in clinical trials. There's one that has emerged with European approval, not yet FDA approval, at $1.3 million per dose, making it the most expensive therapeutic in the history of our planet. So how does this bode well for the kind of disruption that we associate with technology like computer technology? And for that, you look at an adjacent plot, an adjacent observation, which is the cost of reading and writing genomes, and in fact, reading and writing all sorts of technologies, as Daniel mentioned, are coming down exponentially. And they're not just coming down exponentially the way that computing is coming down, they're coming down faster than that, which is incredibly fast. We've seen cost of genomics decrease by 3 million-fold in just a few years, most of it within an 8-year period of time, even though genomics as a field is much longer than that. And I think once we put our mind to it, once we put the engineers on it, that slope changes. For most of the history of genomics, we were a bunch of biologists kind of bumbling around, turning the crank faster, as fast as our pipette men could handle it. And then after the genome project was over, we started a $1,000 genome project. The NHGRI at NIH did this. And then suddenly, for much less money than the genome project, we brought the cost down from $3 billion to less than 1,000 now. And what does that mean about disruption? So when I was, a few years ago when I was teaching a course on computational biology, one of my students went off and bought herself a PCR machine and diagnosed herself based on family history of hemochromatosis. Very inexpensively did this at a time where the prevailing wisdom was that to do a genetic diagnosis you had to have physicians involved, and you needed to have family history, and you needed to have $4,000 per gene. Fast forward to today, and ever since then, basically, people have been figuring out ways to get their genomes done for free or inexpensively, and it will come down in the same way that Google Maps and other software is free. What about therapeutics? So when I think about disruption in therapeutics, I ask myself, well, how many of people that I know are manufacturing their own therapeutics? And I know of 3 different gene therapies that individuals have made themselves and injected themselves with. I'm not recommending this, by the way. In fact, I discourage them. But here is the most expensive drug in the world, or the class of drugs, gene therapy, and now 3 instances, 3 different ones that are done, not just off-label, but completely off the grid. I hope it goes well, because we have a history in gene therapy where very professional teams resulted in a few deaths. One due to overactive immune response to the adenoviral vector of the patient Jesse Gelsinger, and then another set where the gene therapy DNA inserted itself and activated the LMO2 oncogenes. But I think we're in a more advanced state now, and these people that are doing do-it-yourself gene therapy are optimistic, to say the least. So what happens when we start having do-it-yourself reading and writing of human genomes, I think, is the question about disruption here. And even if it's short of that, I'm fully of support of the FDA. I think it's the right way to go. But what is there going to be that kind of disruption in this field? And I'll just close on saying that I've already mentioned the cost of lowering costs is actually remarkably inexpensive. And the cost of prevention is really what we should be talking about. And most of precision medicine, I think the emphasis will be on, even still, on small molecule drugs, despite what Daniel and I might say. And it will be on reactive, precise but reactive medicine. I think usually a tiny fraction of the pie is set aside for prevention, but I think it's extraordinarily powerful. If you look at cancer, most of the things that really buy us 30 years or more of life are preventative. The things like preventative surgeries, if you have a genetic predisposition like BRCA1 or 2, avoiding environmental carcinogens like smoke, and getting vaccines like HPV. So I think if we're going to be disruptive, it's going to be through radical changes in cost and prevention and possibly empowered patients.
All right. As usual, George does not fail to jolt me awake. Three individuals who self-administered do-it-yourself gene therapy. Is that what I heard?
Right, but I forgot to mention, I don't know anybody who's manufactured their own small molecule drugs, although I'm sure they're out there. That is to say, new drugs. I mean, there are plenty of people who make old drugs. Yeah.
Yeah. Okay. Well, that's definitely the brave new world, George. Start with you, Ed. You did talk about the advantages of having a large academic system supporting you. But I'm going to ask you again, what's been the biggest downside of being in a traditional academic health system?
Well, I don't like to dwell on the negatives. There's so many opportunities to do that.
By virtue of what you've done, you've clearly shown you're an optimist.
Yeah. Even when it comes to the FDA, I've had tremendous interactions with them, and they've all been enormously positive. But, a large academic institution carries with it, no doubt, the burden of the momentum and, the inertia that goes along with all those various offices. But you can also use those, the sort of the diversity that institution offers to your advantage as well. I do believe that the leverage we've been able to make with or leveraging the potential of a place like BU, and it's bigger even than big BU, right? Because as has been mentioned here before, what really I think the success that we've enjoyed up until now is really a function of being in Boston. Right, so we've leveraged the interdisciplinary community of this great city, right? We've got the Massachusetts General Hospital down the road, we've got the Joslin Diabetes Center, and we have the med tech industry around it. But BU is a big part of that microcosm, an important part of it to me, because that's where I am. And it is, it's a lot of bureaucracy, there's no doubt about it. But if you do distinguish yourself well in these various areas, you can get the university to be very responsive. And they respond to the very things that we are used to. You raise a lot of grant money, you go rise high on their radar. You publish in high-profile boutique journals, and they get excited about that. You bring a lot of publicity to the institution, and they get behind you as well. So there's no doubt that the success that certain individuals have is amplified in a place like this. And those that don't have quite that visibility, I think, face much more frustration than some of the others. But I'd like to emphasize, the positive side of it, and that is that, it's got an Office of Tech Transfer, it's got, General Counsel's office, it's got, all these great research institutions in the U.S. have these kinds of capabilities, and it's just a matter of using them all to their potential. And we used the office, believe it or not, of sourcing and procurement in a way I think no one has done before at that institution or many others I could think of. In order to build this technology, we used their established, sort of working within their own rules, You use that office to buy office furniture. I used that office to build a class 3 medical device. And they had research relating consulting agreements that they used to set up relationships with vendors. I did that to set up relationships with contract manufacturers, and I paid them through private giving. The university set up for me a little account that I could fund, took no institutional overhead on gifts. That allowed every dollar that was donated to go toward this cause. I could tell that to people when they donated. People like to know that 100% of their money is going directly to that cause in a very focused giving opportunity. And on top of that, it's a 501(c)(3). You can use that to point out that the institution is giving you a tax advantage. So we use the institution in all kinds of ways, and there's a synergy that comes from all those offices if you can get them to talk to one another so they know that each of these various offices is working together to make this happen and sort of buy into it. So again, I don't want to emphasize the negatives too much because I think the positives far outweigh it. Leveraging the university's resources, I think, is extremely powerful. And I don't think academics have done a good job of that. And the academics are on the front line of this. They are the ones that really need to take that leadership role and figure out how to get these universities to engage more in the translation process. Academia has much more to offer, can translate technologies much further than it even knows. The institutions within these, the offices within these institutions don't realize that they should be taking these technologies further than they do before they spin them out. And I just think that these fledgling med tech companies are often suffer from not having resources that they could have benefited from if they kept the technology in-house a little bit longer.
So, Alison, you, I think, correctly referred to the maverick surgeons who did these amazing breakthroughs. But as you pointed out, those surgeries have now remained as they have been for many years, for decades. And so you're proposing some actually relatively incremental changes to some of those surgeries and some are not so incremental. Are there maverick surgeons who are saying, sure, that makes sense to me, let's jump in? Or is no one yet willing to take that act of faith, leap of faith that you were referring to?
Yeah, so I would say, well, first with regard to the progress, I think that at the beginning of the history of developing heart surgery, it was in a way easier to make progress because the conditions were fatal and they had very little to lose. Now that we've become better at treating them, it's a higher risk to change course, and so it's understandable that people are willing to take less risk now. At the same time, we also have more regulations in place, so people are not allowed to try things that they may have been able to try in the past. So we kind of view simulations as a way to disrupt that process and accelerate the process and provide a way of testing things in advance of, testing ideas that are too far out of the box to go directly to the clinic. We have, in the one case that I showed where we were able to take something into the clinic, I would say it was enough of an incremental change that the surgeon was comfortable going ahead and just trying that. And we had some back and forth about the data, and but in the end, he was comfortable enough. In the second example that I showed, it's a pretty drastic change from what they do now. We definitely have clinicians who think it's a good idea, but they're all kind of going, no, you try it first, no, you try it first, because a lot of them feel that their career would be on the line if they try it and it goes south. Maybe even more now than in the past.
So in order to have the Blaylock Talsig Marsden, you'd have to actually have you become a surgeon, is that what you're telling us?
Well, I've got to convince somebody. Yeah.
Right. Very good. And
I don't make enough money to touch the patients.
we could change that.
Just kidding.
I have a quick question for Alison.
Please.
Sure.
These are congenital. How many of them are genetic?
Great question.
So I'm not an expert in the genetics of congenital heart disease. I know that there are genetic factors at play and they're not fully understood. So for example, if you have family members with congenital heart disease, you're much more likely to have, your own children or siblings, but it may show up in different forms in other family members. There's a lot of really interesting work on embryo development and the formation of congenital heart disease. We're involved in some simulations of zebrafish hearts along those lines, but it seems to be a mix of genetics and other factors that are not fully understood.
So
There is potentially also a mechanics factor, so there's a sort of no flow, no grow hypothesis that if you, for example, have a stenosis or narrowing in a valve and you therefore don't get flow to a chamber, then it can lead to a lack of growth, and so there are sort of mechanobiology factors at play.
So Daniel, MIT has a great culture of engineering, but doesn't have a medical school. How does that influence, curtail, or not influence at all your progress?
Yeah, that's an interesting question. I mean, I think the short answer is that we are so lucky to be across the river to the greatest and most powerful medical institutions in the world, in my opinion, with the Harvard complexes, and have developed deep relationships with Harvard, for example, through the Harvard-MIT Division of Health Sciences. So I have some ancillary appointments at some of the Harvard medical schools. And we also have built up the Institute for Medical Engineering and Science that's really trying to further develop bridges to the institutions at Harvard and build medical expertise along with engineering expertise. So I think for me personally, interacting with clinicians has been a tremendously powerful part of the work that I do, and while they may not be next door, they are close enough.
Thank you. So George, where do you see the future of gene editing, genetic engineering going in terms of regulation? Is it going to still look more or less like drug development, or is it going to look more like device or even surgery in terms of regulation? Because conceivably, every different edit is a different intervention, not one drug.
Well, I think it largely depends on how it goes. I mean, it could be none of the above. It could be like vaccines, where every year you get a new seasonal flu vaccine, and it's actually a fairly minor regulatory hurdle to go through. If gene therapies are highly predictable, and I'm not saying they will be, then we can look forward to something like that. We should also prepare ourselves for the possibility that some of them will have unintended complex systems biology effects that have nothing to do with the means by which they're delivered or the means by which they act at cleaving or modifying base pairs. But I have very strong opinion that there will be something highly disruptive here, and it may be not therapeutics, but it could be diagnostics replacing therapeutics that is disruptive to therapeutics. And
Could you give an example? I have no idea what you're talking about.
Wow. So, for example, Alison mentioned that 1% of people have congenital heart disease. About 4 or 5% of people have very serious genetic diseases that are the result of the parents being healthy carriers. And those children, for many of them, fortunately, there are now therapies, just as Alison said, that there are now ways that we can slog along with the congenital heart disease. For many of these others that are individually rare, but they're collectively common, sort of in this 4% range. And those therapies cost somewhere between $800,000 and $30 million per lifetime. The better they are, the longer you are taking them. And so that's why it's not totally surprising that $1.3 million is the cost of a gene therapy. Anyway, my point is that, I seem to be making the therapeutic argument more and more here, but the alternative is for these carriers is if they knew that they were carriers, they, in some societies, in some parts of the world, that who they date is determined by the carrier status. And it doesn't really exclude that many people. It might, you know, if you have a rare disease, the chances are you're not going to be excluded from anybody. But you will eliminate this 4% burden. And then other people are given, they will choose to have abortion or in vitro fertilization. For people who are pro-life, even in vitro fertilization is not necessarily an acceptable strategy, because you might have 10 embryos for every one that's successfully implanted. So it's not really saving embryos. But then something that sounds at first more radical, which is germline gene therapy, could actually be more acceptable, ironically, because if you change the sperm, you could eliminate the carrier status effectively of the father and put no embryos at risk. And so there's some dramatic options here that may or may not, that you know, with just genetic counseling, you can avoid any of those therapeutic strategies.
Well put. I'll give a chance for the audience to ask questions. Are there any questions? I recognize, yes, please go to the, yes, Ricardo, please. Identify yourself.
Yes, Riccardo Bellassi, University of Pavia in Italy. I have a comment and a question. The comment is that I'm really pleased that there was this panel today, because we also in Pavia are trying to work on the issue of combining engineering with medicine in a way. We have just started a Center for Health Technologies, how we have called it. It is an interdepartmental initiative where we are trying to put together the people that are working towards this direction. So, and in the discussion, we were exactly talking about, well, on the one hand, how to train the people, and on the other hand, what could be the most disruptive technologies that could be put in place. One of those that we are working on is the 3D printing. So the question is, starting probably from Alison, if you are actually using 3D printing too for moving from the simulation to the physical model, and in general, if you think that 3D printing, in terms of lowering the cost of some of the devices that can be reproduced also, let's say, at the patient's home, you think that it's a potential disruptive technology?
Yeah, thank you. I probably shouldn't give too strong of an opinion because I don't consider myself an expert. By coincidence, though, my direct office neighbor is one of the founders of 3D printing, Mike Sema. And one of the things that I've learned from him is just how broadly 3D printing can be used, and often in ways that I don't fully appreciate. In medicine, for rapid prototyping, there are efforts to 3D print organs, to 3D print specific bone tissues that map directly to the site of injury, to 3D print tools that you may really not be able to make very easily using other techniques. I think the sense is that it is a very powerful technique. I know it's also generated a lot of revenue for MIT based on the patents, by coincidence, which is some benchmark for commercial impact. So I guess that's my answer.
Alison?
Yeah, so we do use 3D printing to some extent in our lab, and I also have colleagues who've used it, for example, to actually print models of patients' congenital heart defects that they and their families can hold in their hands. I think at times that has been a little bit overhyped in the media, but there are also some nice studies showing effect of 3D printing or virtual implantation, in device selection and things like that. So we use it mostly for experimental models and sort of for educational purposes.
And we use it in a more mundane way. I mean, we use it to build, prototype devices, but it's an extremely useful tool. Again, it's another one of these university resources that would otherwise carry a lot of overhead with it that we get to use basically, or have been using for free as an academic. But it just makes for better devices from the standpoint of sort of the traditional way of thinking about it. you can consider more configurations of the system rapidly and reject designs that have fatal flaws quickly and easily.
I think it does bring up some FDA-related questions in that if you think about 3D printing a custom device for individual patients, and I think that is the direction that things are going, you know, how do you approve, I guess you approve the process, but those devices may be different, and that brings up regulatory questions. For example, everybody has a different shaped aneurysm, and all of those devices you might print for that are very differently shaped and might have different effects on flow, et cetera.
Sounds like the FDA is going to have to get even smarter. Elaine, could you introduce yourself?
Sure. Elaine Collier from National Center for Advancing Translational Sciences at the NIH. And this is a very interesting panel in this week, which is the National Week of Making, June and Makers. And obviously, I think people know about the maker movement, the DIY community, and the question I have for you is, at least one of you probably had somebody with no degree involved, your son, in the design and project that was developed. But how do you see bringing in these other expertise, the tinkerers, the innovators, the people who have something to contribute to the innovation and this disruption for health into this, and how can we do that within our academic and government communities?
Well, the do-it-yourself community has been tremendously impactive in automated glucose control over the past couple of years. The nice thing is you don't have to bring them to the table. They inject themselves, right? They don't take, there's a hashtag out there saying we are not waiting, and this has to do with getting technologies to people's kids who have type 1 diabetes, because the cure that Doug Melton, that we're all hoping for, will ultimately bring to the fore is a couple of decades out, I would expect. As optimistic as we could be, I don't see it much sooner than that, because it's not just making islets by the bucketful, it's solving an autoimmune disorder. So replacing the beta cell mass doesn't solve the problem, that will immediately be eradicated. So the challenge is, of course, a much bigger, broader one. So until such time as the cure is available, we need to build these technologies, and that's what the do-it-yourself community has been doing. It's a very small subset, of course, because there's a lot of skill in the technology space to build these devices, to make little systems that can communicate between a continuous glucose monitor and pumps and a smartphone and an app that you would develop to run on a smartphone, and a little controller that operates all these components. It's not something you can it's not easily done in your basement, but it can be done in your basement. What they've done by virtue of their persistence is hugely important, because they've sort of put it in the face of the industry, saying we don't really need to wait for you. Now, it's not something that scales. It's not something that more than a few thousand people have been able to take advantage of. And it is something, by the way, that the FDA has been very tolerant of. They really have sort of looked the other way. But that's because it's been limited to a few thousand people. But nevertheless, they have a loud voice, and it's a good thing in terms of the impact it's had on driving the industry to recognize that this is something people can do themselves, or a small subset of people can, and we are good at scaling, or not we, meaning the med tech industry and the pharmaceutical industry is good at scaling. That's what they do well. They don't do a lot of things well, but they do that well. And so it has, I think, spurred, at least if not innovation, incentive to to integrate their technologies. They're beginning, the DIY community's begun to get the various companies, each on their own platform, that there's no talking between them, right? They all have their own different protocols. There's no standardization. Get them to the table to talk about standardization of different platforms so that we can make these things interconnected and allow patients some choice between different technologies. Some technologies work better for some people than others. Having choice is very helpful, and the do-it-yourself communities can be very helpful in bringing that kind of integration, that kind of uniformity across platforms. So I just think they're invaluable to pushing the field forward and pushing the envelope.
One way that we've incorporated that is through open source software. So We haven't had a lot of contributions to the source code from outside a select few groups, but the source code is available publicly and people are welcome to contribute to it. We do have users from all over the world, probably every continent of our software for a range of different reasons, from research to education and beyond in industry.
Introduce yourself, please.
Yes, it's Ned Bacon. I have a question for George. I was interested in the 4% number you threw out, and I think you used the word burden when you were describing that. And my question is, we've been evolving for a long time, and I would that strikes me as a very large number to be remaining at the current point of evolution. And we're now at a point where we've introduced this feedback into the evolutionary process, which becomes very powerful. My question is whether the fact that so much of that is left over, that burden so-called, is that an unfortunate artifact that's left, or is there some evolutionary advantage there that we don't understand that maybe there's some diversity remaining, there's some robustness that we'd remove in taking that on?
Just to make sure, your question is about why do we have recessive diseases? Okay. Yeah.
Are we going to be removing overdominance advantages, for example?
Right. Yeah. So I think the effort is not really to remove the alleles, the variants from the population. It is to remove the disease. So, for example, if you have two healthy carriers, 1/4 of their children are heavily diseased, 1/4 are not carriers, and 1/2 are carriers, and those half are fine. And to first approximation, there's no real public health or even individual effort to get rid of those carriers. For dominant diseases like BRCA1, where the mother has had the experience of having surgery preventatively, there are some mothers that are doing in vitro fertilization, PGD, so that their daughters do not have the allele at all. But that's a different case. For the larger set of recessives, I don't think we would be changing the population frequencies, because there are heterozygote advantages, many of which we don't even understand. And I think, if anything, we want more diversity, not less. And so far, we've paid the price of having these horrible homozygote diseases. But I think we now may be getting into a win-win situation where we can avoid those. Tay-Sachs, for example, just by selecting your marital partners, has been almost eliminated from some of the populations of the world, like the Ashkenazi population.
Gentleman from Novartis, introduce yourself.
Yeah, at Conclusion Novartis, I have a question for George about future of gene editing. So in drug development, we have a concept of druggable genome. Is there a concept of editable genome? And if not, then do you foresee ever tackling polygenic diseases, sort of fixing several things at a time?
That's really interesting. I'm surrounded by people who talk about druggable proteins and undruggable. For example, my company, Warp Drive, we talk about this a lot, and believe that we have now solved the undruggable protein problem, but that's a separate issue. I haven't heard that referred to with uneditable. Certainly, highly repetitive genes were thought to be difficult depending on your goal. If your goal is to get one of them, then you would have to somehow have your targeting dependent upon something that's outside that is unique, and that's certainly within the realm of possibility. We recently had a very pleasant experience with hitting repetitive genes, where we wanted to target all of a family of retroviruses so that we could make pigs that could donate organs to humans without risking retroviral transmission. and we got rid of all 62 instances of a gene at once with essentially with one therapeutic dose over a 14-day period in cell culture. So that would be my first reaction to your novel question, is that maybe repetitive elements would be a little more challenging. But I really think, the first pass is going to be focused on restoring rare alleles that are deleterious to their wild version, and that's, in a certain sense, we've already done the experiment that those work pretty well. And then another set will be targeting pathogen mechanisms to hit it. So, for example, knocking out the receptor for the HIV virus, CCR5.
So last two questions. Introduce yourself.
Hi, Christine Chen-Silvers, actually trained here and at MIT, and Chief Medical Officer of Health Reveal. I actually have a two-part question, but...
Go for it, go for it.
So the first part is towards precision medicine. If we have successful clinical trials, that we get these insights that the number needed to treat for such and such therapy is 20 or 30. Do you have some thoughts on how we can try to reduce these numbers needed to treat towards 1?
I don't understand the question.
Well, if you have a great randomized controlled clinical trial
Yep.
and they discover that, using this therapy in 30 people or on average every 50 people, you're going to save 1 stroke, you're going to save 1 admission for heart failure, you're going to save 1 death, and so on. So are there some ideas in terms of engineering and technology how we can be able to get these therapies that are proposed to have a number needed to treat as close to 1 as possible, so that you don't have the other 29 people who are getting the therapy without any benefit or with possible harm?
Got it.
I mean, I think that's very much dependent upon the technology you're talking about. The device that we have, the number to treat is 1. That we're there with this device because the effect size is so large. So we are essentially nearly normalizing glycemia in everybody on the device, and the standard of care is so bad that the effect size is large by virtue of 2 things. The device works really well, and the standard of care is really bad. So that disparity helps you achieve that. But it's really going to depend upon, the answer to your question depends tremendously on the technology and the circumstances of the chronic illness you're trying to treat.
And then the second part was, let's say if we use big data and machine learning to learn various insights to try to get towards that goal of a lower number needed to treat, do you think that we can just use those insights that we learn from machine learning, or do you think because of its statistical correlation nature that we need to then vet those out in clinical trials?
I think we're going to be talking about some kind of clinical trials. We might be able to bring down the cost of those trials by precision medicine, but I think that it would be unwise to completely sidestep them. Some of these things that look like correlations now may turn into cause and effect explanations with very complicated causes and possibly complicated effects. Big data and deep learning may get us to those causes. But also laboratory models and clinical trials will be part of the deal as well.
Great, thank you. Now our last question.
Thank you, Zach, and thank you all of you for another fascinating panel. I want to come back to the topic on the board, will engineering disrupt therapeutics, and talk a little bit about the process of innovation. My question has to do with the international contributions to innovation. Alison addressed this a bit when she said that her software is being downloaded, open source software is being downloaded by people worldwide. I guess I'm more interested in the process of coming up with new devices, of actually disrupting therapeutics, coming up with medical devices or coming up with software protocols that can actually help people. To what extent do you see, panelists, the contributions towards this type of innovation coming from outside the United States? And I don't mean Western Europe and Japan, I mean really like the developing world. And I ask this in the context of a piece I wrote a few years ago for my blog, Boston Biotech Watch, where I observed that a lot of innovation arose from people taking ideas or thoughts from here, going to environments which are not as heavily regulated in India, for example, Asia, even in the Caribbean, and doing procedures at lower costs, practicing, as it were, and coming up with ideas that could then be translated back. So there are companies around here that are actually working on, paper-based diagnostics and tools like that that can really be helpful in the developing world that also can create business models here. I realize it's a bit of a loaded question because I'm coming with an agenda. On the other hand, I thought perhaps because you are on the forefront of innovation, you might have some experience with the way that the feedback loop is working. Is it really just, when we say engineering, MIT and Stanford engineers who are out to disrupt therapeutics, or is it coming from other places?
Good question. George, want to take a whack at it?
I'll take it. So I do see some innovation coming out of many developing nations. Some of those really wouldn't, but I think there's a bit of we're kind of glorifying simplicity, not necessarily as disruptive as complex things are. So for example, in many developing nations now, you have a lot of cell phones, but those were not simple paper-based electronics. They were very complicated things that we brought the price down. I think you can often find a lower price. So one of my climbing buddies, Jeff Tabin, had a clinic, set up clinics throughout the Himalayas to treat cataracts, and obviously the cost of the lenses alone were prohibitive, and he would train a village by going through. He would do one eye, and the barefoot docs would do the other eye. And at the end of the day, there were a lot of people that were blind in one eye, but there were also a lot of people that could see, and they would manufacture the lenses for about 1/500th the cost. Now, I wouldn't say that's necessarily a totally transferable back to New York City, but it's the best I can come up with. But I really think we should also applaud really complicated things that bring costs down radically.
Great. So with that, I want to thank our panel